Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. And we are about to start our biomedical engineering session. And welcome everyone to this exciting session. And here we have a session that is for the men and the women, and we have both panel speakers. I will be moderating on the women's side, and I have my co-moderator, Junaid Malik Sahab, who's engineering professional, will be moderating on the men's side. So with that, I would like to briefly introduce myself. I'm Dr. Nusrat Sharif, and I have over 16 years of experience in large pharmaceutical industry, Pfizer, Bristol Myers Squibb, and now at AbbVie. And my field is immunology. So this is a really interesting venue where we are going to explore some of the recent advances in biomedical engineering. And we have 3 exciting talks that are lined up in this session, bringing different aspects of biomedical engineering and blending them towards how we can achieve excellence in medicine and in driving patient care. So with that, our first speaker is Aziza Faruqi Saiba, who is an enterprise architect and has a passion for digital transformation. And she will be talking about how digital transformation as well as emerging technologies are transforming clinical trials. And why clinical trials are important? Because the medicines that the patient receive, they have to go through the rigorous process of clinical trial development before they are approved by the FDA or regulatory agencies. So Aziza Faruqi Saiba will be talking about how we can leverage the clinical trials and accelerate them by leveraging the AI technology. And with that, I would like to welcome Aziza Faruqi Saiba. Our next speaker would be talking about neoantigen vaccines and the role they play. So cancer treatment has been pretty challenging, and even the latest advancements are still not completely giving care to the patients in need. So there is a significant unmet needs, and neoantigen vaccines, where you can harness the immune system to fight the cancer-producing cells, is a great way in the biomedical approach. And so we have a young biomedical scientist here who will be talking on that aspects of developing new antigen vaccines for anti-tumor immunity. And the third presentation is by bringing or merging mathematics, engineering, and medicine to bring non-invasive technological advancements for patient care, particularly in cardiovascular diseases. This particular talk will address how this novel technique of electrocardiography will monitor the movements of the body and measure the cardiac function. And this is particularly useful to monitor because it is non-invasive, and there are sensors that the patient can have at their bedside or wherever they are that will be monitoring their cardiac function, and can be easily used to determine whether they are having a cardiac disease or whether there is improvement in the intensive care unit. So early diagnosis of disease as well as enabling monitoring of a patient with a cardiac disease is especially helpful with this. So with that, I would like to first welcome our first speaker, Aziza Faruqi, and I would give the panel to her to share her exciting talk on emerging technologies and AI for excellent clinical trials.
Assalamu alaikum wa rahmatullahi wa barakatuh. Jazakallah for the introduction. In my presentation today, we will explore how emerging technologies are transforming drug development process and accelerating clinical trials. In early 2020, the world stopped. Borders closed, hospitals overflowed, and families faced a terrifying uncertainty. But at the same time, a miracle happened. In under 1 year, we developed, tested, and deployed multiple vaccines, a process that normally takes a decade. The pandemic showed what's possible when science and technology works together. And it forced us to ask the question, if we could move this fast for COVID, why not for cancer? Why not for ALS? Why not for heart diseases? So today, I want to take you on a journey through the clinical trial process and show how emerging technologies and AI are reshaping it. Not just to move faster, but to move fairer, smarter, and more humanely. But first, to set the stage, let me introduce you to clinical trials. Drug development starts with the discovery of a promising compound by scientists, followed by preclinical trials in the lab. And then we move on to the clinical trials. Phase 1 of clinical trial is to test the safety and dosage. It's run on a small number of healthy volunteers. It lasts for 6 to 12 months. Phase 2 is to test the efficiency, side effects, and to fine-tune the dosage. It's run on several hundred patients, lasts for 6 to 12 months. Phase 3 is to confirm the results that we found in the earlier phases. It's run on thousands of patients. Comparison is also made with existing treatments, and it can last for years. And phase 4 is post-marketing. It's run after the drug has gone through the approval process, and it is to test the efficacy of the drug over time. And we gather real-world evidence during this phase. Now, let us walk through why clinical trials are a bottleneck. Clinical trials are expensive. Basically, the drug development process itself is very expensive, and it can cost up to $3 billion per drug. And most of that cost goes towards running the clinical trials. They have long timelines, and they can typically run from 10 to 15 years. 80% of the trials are delayed due to recruitment challenges. They're subject to complex regulatory requirements because the drugs themselves are very complex now. 90% of the clinical trials fail to achieve results, meaning that they are not approved by FDA or any regulatory body. Lack of high-quality data results in analysis challenges. And these clinical trial inefficiencies delay life-saving treatments. Now, because we have very short time, I will limit our discussion to clinical trials on medicinal products or drug products, and we'll stay within United States. I've also divided the process into 3 stages. First stage is the trial startup or the study startup process. Stage 2 is the clinical data collection and management of that data, and stage 3 is the conclusion or submission to FDA. Now let us embark on that journey. Now, protocol is the blueprint for a clinical trial. It lays out how to test the hypothesis from preclinical research. It defines every aspect of the trial, from eligibility criteria to treatment methodologies to what success looks like. Developing effective protocol takes months. Emerging Technologies Toolbox offers multiple tools to expedite this process. Artificial intelligence, AI, and machine learning, ML, these tools perform complex analysis and research. Digital twins and virtual models help us run simulations, and generative AI or Gen AI using large language models or LLMs becomes a medical writer's best friend by generating protocol drafts very quickly. After finalizing the protocol, the sponsor submits the investigational new drug application to FDA seeking approval to begin the human trials. Here again, GenAI helps to compile all clinical and non-clinical content in a standard electronic common technical document, or ECTD format. FDA has 30 days to review the application in minute details before making a decision. While FDA already has been using AI, it recently announced ELSA, an AI assistant, to speed up the review process. ELSA has been in the news recently because, according to an FDA official, it hallucinates confidently. But we have to keep in mind that ELSA is in its early stages, and it will mature over time. Now, once FDA approval has been granted for the clinical trial to proceed, the sponsor must select sites to conduct the trial, finding clinics that are compliant, experienced, and can recruit diverse population is a slow and costly process and is usually outsourced by the sponsor. Here again, AI is used to analyze real-world data and site history to identify high-performing sites with advanced infrastructure. ML can even predict if a site will meet recruitment goals. Now we get to the most challenging part of clinical trial, which is the recruitment. Even the best trial design fails without the right participants. A whole plethora of emerging technologies is used to address this challenge. AI mines electronic health records to cut down the patient matching time, while GenAI translates informed consent forms in patients' languages to make enrollment more inclusive. Digital health technologies like the wearables, like our Apple Watches, telemedicine, all enabled by the advancement in broadband, helps retain patients through decentralized trials. These technologies get us to the FBI milestone much faster and more efficiently. An FBI first patient in milestone is when the clinical trial can start. Now we get to stage 2 of clinical trial. Clinical trials gather large volumes of data from many sources. First, it has to gather data collected at the sites. But that is only 30% of the clinical data that is collected during the clinical trials. It also gathers lab data. It also gathers, it has to collect image data like the X-rays and other images. And then variables. Now our Apple Watches are also sending it data, and other monitors that are used to capture real-time data, that is also being used. That is also sending data. Electronic health records, which is the real-world data, that is also being collected. And then because FDA has a requirement that any severe adverse event must be reported within 24 hours, adverse event data is also coming in. Smart integrations consolidate this data into data hubs. Cloud platforms make this data available in real time for global collaboration, validation, monitoring, transformation, and AI-driven analytics. This is where I would love to be in Dr. Rafi's session, because he's talking about using AI in image reviews, right? And if you miss it, I would definitely watch the recording. Now the last, now before we move on, statistical analysis is key to making sense of clinical data. Data scientists use AI and ML tools to gain deeper insights faster and more accurately. And now even quantum computing is being used to run complex compute-intensive analytics. Here again, digital twins are used for predictive analysis. And now we are ready to submit a new drug approval request to FDA. AI is accelerating the process on both ends. Sponsor is using GenAI to compile clinical, preclinical, and safety data into submission packages. And FDA is using AI tools to accelerate the review. And then after FDA approval, the drug is manufactured at scale. It's distributed commercially to pharmacies and healthcare providers. Real-world evidence is used, from its use is continuously collected to monitor safety and effectiveness over time and to support phase 4 trials or for any future regulatory updates. Now, to summarize, we are in a new era of clinical trials powered by technology. A fast expanding technology toolbox has many tools to accelerate the process. AI, ML, and agentic AI, which we didn't even discuss because we didn't have time, for faster analysis and insights. LLMs and Gen AI for faster content generation. Cloud platforms for scalable and secure data sharing. Data hubs and advanced integrations for frictionless flow of data. Digital twins for virtual simulations and predictive analysis. DHTs or digital health tools for continuous patient monitoring. And decentralized trials via telemedicine. But we must not forget that technology is here to accelerate, not to replace us. It is human plus machine model that drives real impact. Machines alone will only result in chaos. Now I want to close on a verse from Holy Quran. Allah Ta'ala says, "And whoso gave life to it, it should be as though he had given life to all mankind." So this isn't just about making things faster, it's about saving lives. And I close on a quote by George Merck. "We try never to forget that medicine is for people, it is not for profits." That is not just a slogan, it is a responsibility on us. Jazakallah.
Thank you, Aziza. That was a very enlightening talk and gave us good insights on the role of AI and ML. While we have a parallel session of AI going on, you could be a good fit there as well, but tying it with the biomedical engineering gets a unique blend with patient care here. So thank you for that. And I also just want to mention that for the question-answer format in the panel discussion, we'll hold it to the end, as like we saw in the previous session. And the audience can have their questions or even write them down. So at the end, there'll be a panel discussion where we can have question answers answered by the panel speakers. And with that, I would now give the mic to my co-moderator, Junaid Malik Sahab, to introduce our next speaker.
Assalamu alaikum. Our next speaker is Salman Ahmed, Dr. Salman Ahmed Sahib. As Dr. Nusrat already gave a detailed introduction. Salman Sahib was trained as an aerospace engineer from Purdue University, then medical school at University of Wisconsin. He trained in general surgery at Maine Medical Center and surgical critical care at UC Davis. He works as an acute care trauma surgeon at the University of Missouri since 2013 and as an associate professor. So I welcome Dr. Salman.
Jacqueline. Okay. Thank you to the organizers for inviting me, most importantly to my wife, Dr. Summer Ahmed, who's been organizing this as well, and she finally convinced me to come this year. My talk will summarize a non-invasive hemodynamic monitoring paradigm that leverages a mathematical model of the human cardiovascular system. A'udhu billahi minash shaitanir rajim. Bismillahir rahmanir rahim. From the Holy Quran, chapter 50, verse 17. I think this quote manifests what we're really going to talk about for the next 10 minutes. "And assuredly we have created man, and we know what his physical self whispers to him, and we are nearer to him than even his jugular vein." So the context of our research and this talk is the global burden of cardiovascular disease, be it heart failure, myocardial infarction, hypertension, stroke, and other peripheral vascular disease. Clearly a major cause of death around the world. It also affects men and women separately. While men may contribute to 1 in 3 deaths over the last few years, in 2013 at least 1 in 4 women succumbed to cardiovascular disease. And unfortunately, in women in particular, it is very insidious in that it has very little antecedent symptoms, and it often comes on without warning, resulting in sudden death. Clearly, early diagnosis is the best weapon we have for treatment, and we reserve medications and surgery as a last resort. So in that context, some of you may recognize what you see here as some of our older, more traditional tools that we use to diagnose cardiovascular disease. So there's the EKG, the electrocardiogram. There are more invasive methods like the pulmonary artery catheter, which we insert into the heart in addition to an arterial catheter that we put into somebody's artery. All of these are very helpful, but also very invasive and have their own risks and benefits. What you see on the right side is what we are talking about, which is a load sensor that we can place under the mattress of a patient, and it can be the patient in a hospital, it can be a nursing home patient in a nursing home. And it basically gives us what we call the ballistocardiogram. So we're going to talk about what that is and how we use mathematics, which is the universal language of the universe, to assess cardiovascular disease without even touching the patient. So what is ballistocardiography? So I'll call it BCG because it's quite a mouthful. Essentially, think about a center of mass, right? So if you have 2 objects that are of the same mass, the center of mass is right in the middle, as evidenced by this fulcrum. But if one of the masses is bigger than the other, if that changes, that center of mass changes, right? We already know that. So now imagine, as the center of mass changes, if this happens in real time, you now have real-time changes in the center of mass, and the fulcrum has to change in position. So think about this in the context of your body. Your body's center of mass, even though you may be standing or lying completely still, is always changing on a very minuscule, almost imperceptible manner. As your heart pumps blood, the counter motion of the heart's pumping, as well as all the blood that's sent throughout your cardiovascular system, induces tiny counter changes in your center of mass, which you and I cannot sense. But we, through the sensor that we use, can sense these changes and actually extract a considerable amount of information that we never could before non-invasively. So this is an example, and I'm not sure the video will work, but that's okay. What you see is one of our grad students in the lab, and he's lying on what we call a kind of a load sensor or a load bed. Essentially, it's a suspended bed, which in theory is disconnected from any external forces. And what the video would show is, if you look closely down at the bottom, there's a little chalk drawing of a 2-dimensional axis, and there's a laser pointer. And over time, without any imperceptible external forces, you'll see that laser pointer move. And that pointer is moving as a result of the counter motion of the patient's own breathing, as well as their heart pumping. So the question is, can we design a sensor that's sensitive enough to pick these tiny deviations in the center of mass up, and can we extract information from them? Let's see. Oh, it might be working. Let's see what happens. So there is some audio, but again, if you watch carefully, and I know this sounds like looks like a parlor trick, but it's not. There actually is movement that we are not doing ourselves. It's the patient's own body, own breathing and heart rate and blood pressure that are moving the bed, which otherwise is disconnected. So of course, we can design a sensor, and that's what this talk is about. That's the BCG sensor, ballistocardiography, where we can design a sensor that can pick up these tiny changes not only in respiratory rate, heart rate, and can even extract blood pressure without touching the patient. And what you see is the EKG tracing, and you see what we call the BCG signal underneath. And that signal you're seeing is just one axis, but in fact, the body has 3 dimensions of motion. Most of it is in the axial or lateral, but there is some kind of anterior-posterior movement as well, clearly not as much, because the heart pumps blood throughout your 3-dimensional system. So this entire premise is using a sensor designed to pick this up. Now, what do we do with that data? So we have 3-axis data, and we combine that BCG sensor data into a MATLAB model, which is designed to reconcile that data with what we call an electrophysiologic model of the cardiovascular system. So if you think of the heart being replaced by a battery or a volt source, and the current being the blood, and then the blood pressure being resistance and capacitance, if you design a system and an algorithm that encompasses the entire body's cardiovascular system as an electrical circuit model, the MATLAB system then reconciles that BCG signal into that model and gives us what you see here. It can give you not just heart rate and blood pressure, but we believe, and we've done this theoretically in the lab, that if we combine it with an audiographic signal of the heart pumping as well as the EKG, and we combine those three signals, we can regenerate what I, in the kind of critical care realm, call the holy grail of hemodynamic monitoring, which is what you see on the lower left corner, which is a pressure volume loop of the heart. So as the heart pumps, right, opens and closes and the stroke volume ejects, we can extract how hard the heart is working with cardiac power output. We can also extract very discrete thermodynamic principles, which is how efficient the heart is. As we get older, our heart gets more inefficient. As we are critically injured or are septic or have blunt cardiac injury in the world of trauma, our heart gets less efficient. So if we are going to treat that heart, it's best to know exactly what the thermodynamic efficiency is before we just throw blind medications at it, because we can change the blood pressure, but unless you fix the thermodynamic efficiency of the heart, you're going to lose the heart and you're going to lose the patient. So, of course, from theory to practice, one of the first uses of our sensor in the real world, was done at the University of Missouri with our engineers using a prototype BCG sensor. We compared healthy subjects, much like one of our grad students here, this is Muhammad, who's actually received his PhD just last year, compared it to real ICU patients, where we basically evaluated the 3-axis BCG signal and compared it to their EKG tracing. And we wanted to see if we had accuracy equivalent to the EKG tracing. So this is, again, without going into the details of how the math was done, we effectively compared the two signals. We regenerated our own BCG signal and the EKG signal. And it stands to reason that if your EKG signal tells the heart to pump, there's a slight millisecond delay in the heart actually pumping. It takes a little bit of time for the heart to fill, pump, and eject for you to register a signal that something's moving and register a heart rate and register a blood pressure. That delay actually we found to be a very discrete variable that we found to be significantly different in people that are ill and people that have heart disease. And we've actually found that difference, which we call the TEB, which is the time interval between the EKG and the BCG, to be different between men and women, even healthy men and women. So we think we have found a non-invasive method, a metric or a variable that not only tells you if you're healthy, but if you're male or female, and whether you have some pathological heart disease that is manifesting in delay between your EKG and your heart pumping itself. So this is just a quick snapshot of that TEB interval across several sessions and several patients. And you look and you see the p-values are not significant at all. But that's the point, right? We don't want them to be significantly different. What this tells you is that across several sessions, we have a repeatable and reliable TEB data from our BCG signal that is not different, that we can repeat this data and we can actually use it across several patients to gather very discrete information about somebody's cardiovascular health. So I'll just close with a picture of what our latest generation sensor looks like. This essentially is a box that looks like about the size of a small child's shoebox. It literally sits on the back of the mattress. If you think on the lower right corner, there's the mattress. That's the head of the mattress. And so the patient would never see it. It sits on the back, and it's sensitive enough to record that BCG signal with the patient who's intubated on the ventilator, sedated, and not moving, regardless of what lines and tubes and drugs are being infused, and generate that same signal with a high degree of accuracy. We're actually using this now in an ongoing study where we're comparing it again to our arterial waveform, comparing heart rate and blood pressure, and eventually combining it with an EKG signal to generate what I call that holy grail, which is the PV loop. So again, I glossed over a lot of things. I didn't want to kind of talk too much about some of the mathematical background, but this is a perfect example of how we took an engineering principle, created a sensor, and deployed it in real time in real patients. And I'm hoping that with more data, we'll get even more kind of experience and hopefully get some funding to do some more fun stuff as we like to do. This is just an example of some of the things that we've done and shown around the country. Some of the work includes the gender differences that I was talking about. And I'm hoping, especially in light of some of the early plenary session we had this morning, that something like this can be deployed in what we in the trauma world call austere environments, where instead of having a complex hemodynamic system, you have something that literally fits in a shoebox, can be plugged into a power source, and give you information that before you needed some very invasive lines to do. So again, thank you for this opportunity. I couldn't have done this without the innumerable number of people working in the lab, both at the University of Missouri, University of Maine, even some researchers in Italy. Foresight is a company that makes the sensor, which is actually licensed out of work done from the University of Missouri. So again, Jazakallah, and thank you for the opportunity.
Jazakallah, Dr. Saab. In the interest of time, I'll pass it back to Dr. Nusrat. And if there are any questions, just hold them until the very end. Jazakallah.
Jazakallah, that was a really interesting talk. And so now we are coming to our final speaker for this session, who's a young undergraduate, completed her undergraduate in Johns Hopkins University, Sabahat Rahman. And she is a member of both the Scientist Association as well as the Medical Association, as she aspires to go forward in MD, PhD. And her interest is in computational oncology and cancer immunology. And so with that, I would like to welcome Sabahat Rahman for her topic, Boosting Anti-Tumor Immunity with Neoantigen Vaccines.
Assalamu alaikum wa rahmatullahi wa barakatuh, respected attendees. A'udhu billahi minash shaitanir rajeem, bismillahir rahmanir rahim. As Dr. Nusrat Saiba so kindly explained, today I'll be discussing boosting anti-tumor immunity with neoantigen vaccines, which are a relatively new form of cancer treatment. To start, however, I'd like to discuss the current state of cancer care and why it falls short. Traditional treatments have been around for many decades, and in fact prior to the 1970s, and many of us are familiar with these. They include surgery used to tackle resectable tumor masses, radiation, where high-energy waves like X-rays are used to shrink local tumor sites, and chemotherapy, where drugs can be delivered to have systemic effects on killing cells. More recent treatments have come out in the last decades and continue to be researched and studied. And these include hormone therapies, targeted therapies, immune checkpoint inhibitors, and others. Importantly, however, the trends in 5-year survival rates really vary across cancer types, highlighting the limitations of these treatments. For instance, we see that with prostate cancer, 97% of patients may be expected, statistically on a population-wide level, to have a 5-year survival rate. However, with something like pancreatic cancer, this number drastically falls, and it's just 13%. This is attributable to a number of reasons. For example, prostate cancer is often detected earlier, whereas pancreatic cancer is often detected at very late stages. And moreover, prostate cancer is more responsive to existing treatments, whereas pancreatic cancer is not. This is where neoantigen vaccines come in and where they may be able to help address limitations of existing treatments. Now, broadly, neoantigen vaccines fall under a category of therapies called immunotherapies, which, as the name suggests, are used to harness the body's natural defense system, its immune system, to kill tumors. With neoantigen vaccines, looking at this diagram on the right of the screen can be helpful, and I'll walk through it mainly in 3 steps to help explain what neoantigen vaccines are attempting to do. First, in our body, we have what are called antigen-presenting cells, T cells, and then antigens. Antigen-presenting cells, first, step 1, take an antigen, which is a small protein fragment, and portray it on their surface, shown here in pink. Then these antigen-presenting cells encounter inactive T cells, and these T cells are really our body's main attacking cell. Finally, when the antigen-presenting cell shows the antigen to a T cell and they bind, a T cell actually becomes activated, and then it's ready to go on and attack cells containing that specific antigen. The key innovation with neoantigen vaccines is this. If our antigen is a protein fragment from a tumor cell specifically, we call it a neoantigen. And if we use neoantigens to prime T cells, we can elicit T cell activity that's tumor specific, essentially using our T cells to specifically go and attack tumor cells as opposed to healthy ones. Now, neoantigen vaccine development really bridges computational research, wet lab research, and clinical care. And I think that's why it's really such a wonderful topic for biomedical engineering, because it does involve a spectrum of disciplines, including BME. On the in silico or computational side, researchers will first analyze tumor and normal DNA to identify neoantigens and candidate neoantigens. Secondly, computational algorithms are used to actually rank these neoantigens and determine which ones are most likely to activate T cells. Then in vitro, or essentially in a wet lab like a biology lab, researchers will test neoantigens in cell culture and actually validate whether or not they activate T cells. Synthesizing a vaccine with these neoantigens is also a critical step, and often there are additional components. These may include, for example, adjuvants, which are used to further stimulate the immune response. Finally, in vivo, neoantigen vaccines have to be tested in preclinical models, for example, in mice, and then, of course, progress on to clinical trials. So you may be wondering if these neoantigen vaccines work, and there really is a mixed body of research on this. The first clinical trial was held in 1997, and since then there's really been a flux of phase 1, 2, and even 3 clinical trials for these neoantigen vaccines. Across the board, what we see is that these vaccines tend to be safe and induce minimal side effects. And this is really promising because a huge lacking with current cancer therapies is that they induce many adverse side effects and promote nonspecific killing of both tumor and healthy cells. So with neoantigen vaccines, the fact that we see minimal side effects is a promising sign. On the other hand, if we're thinking about efficacy, how well neoantigen vaccines actually target tumors and shrink tumor masses, for example, results are mixed. Some patients respond very well, whereas others don't, and this can even be within the same tumor type and the same patient cohort. Therefore, there's really been a lot of interest in actually trying to characterize patient-specific immune responses and determine which immune characteristics may make patients more or less susceptible to these vaccines. Finally, I'd just like to end by really emphasizing that neoantigen vaccine development is an evolving and very active field of research, both on the in silico side as well as on the in vitro and in vivo side. In silico, there's a lot of interest in building better algorithms to rank neoantigens because current algorithms do fall short. They identify very few true positives, so in other words, very few neoantigens that really do elicit T cell activity. In vitro, screening candidate neoantigens is a major task, and this is because it's very both labor-intensive and expensive to test hundreds of neoantigens. So figuring out efficient screening technologies is also important. And then finally, in vivo, there are many options for vaccine formulations in terms of what specific materials are used, what specific adjuvants, and so on. So characterizing and trying to understand which vaccine formulations work best is really important. And finally, just to reiterate, a lot of labs across the US are working on this. Alhamdulillah, I have the opportunity to work with Dr. Rachel Karchin at Johns Hopkins, and she's very interested, and we're very interested in the computational side of this problem. So we've been working on algorithms for neoantigen ranking. Finally, I'd like to end and bring this all back to a quote from the Holy Quran. So Allah Ta'ala tells us, Bismillah ar-Rahman ar-Rahim, "And there's not a thing, but with us are the treasures thereof, and we send it not down except in a known measure." And I chose to include this quote because I feel that neoantigen vaccines hold a lot of promise, but there are a lot of unknowns. And I think as researchers, we can humbly sort of think that Allah Ta'ala knows what will make the best vaccine or even the best cancer treatment. And we still don't know, so that's what we're trying to figure out and inshallah someday come to. So Jazakallah. Thank you so much for listening.
Thank you, Sabahat. That was very interesting and kinds of gives us in our mind what will be the future of this cancer care in the next 5 years. How do you see it emerging with the neoantigen vaccines? So that could be one question that came in my mind. But before you could answer that question, I'll also open the floor for questions from the audience. And so we can have a few questions from the women's side, and then I'll pass the mic to the men's side for their question. So my first question, I can begin with what I've asked, the future of cancer care, building these kind of emerging platforms for therapy.
So, Jazakallah, Dr. Nusrat. So neoantigen vaccines, what's interesting is they are a personalized vaccine. So in terms of implementing these in patient care, really one big strategy is sequencing patients' DNA and determining a specific vaccine formulation for them. So I think in the future of cancer care, inshallah, and we're already starting to see it in clinical trials in rare cases, these personalized treatments may come to fruition rather than general therapies. And so developing personalized neoantigen vaccines would be very important. I don't know if that answers.
Yes, thank you. Yes, it would be a time where that could be a routine care. That was my question, where these vaccine treatments would be incorporated, routine care for the cancer patients. And that could be something we can look into the next 5 to 10 years, inshallah. Anyone else from the women's side who have questions? Please feel free to ask your question. I do have one question for Aziza, Faruqi, Sahiba, and that talk was really interesting, and the future of clinical trials. I was wondering if you have a specific example of a clinical trial in your work, or that you could describe, where you saw that the AI meaningfully shortened the timeline without compromising safety.
So I can give examples of how, we talked about multiple AI technologies. GenAI is an up-and-coming technology, like about 2 years, but in the new world of fourth industrial revolution, 2 years is a long time. Right? GenAI is very extensively used in multiple areas. The first, I would say, in protocol development. Right? While AI is doing the analysis, GenAI is actually putting it together in a structured format. Like, then wherever we have any kind of structured documentation, right? GenAI and LLM is really changing the world. Right? On the recruitment side, which is a huge challenge, AI is being extensively used because if you think about it, finding the right patients or the right participants is not rocket science. Is really matching the inclusion-exclusion criteria from the protocol with the patients or the participants who match that. This matching, while it can be done manually, it can be expedited using AI technology. Right? And we see that in recruitment, there is a huge benefit. In fact, there is an AI tool which is developed by NIH, which is for this purpose. And it's actually cutting down on the matching time for patients by 40%. And the bigger benefit of it is that it is making the recruitment and enrollment more inclusive. I don't want to make it a long answer, but while my flight here, I was listening to the Invisible Woman, right? And it's the lack of diversity in our clinical trials. AI is actually helping there a lot, right? because while manually assessing that data is possible, AI, and on top of it, if you put agentic AI also, it is highly expediting that process. So that's a very easy area, an area where we are very visibly seeing the expediting of clinical trials through AI and through Gen AI.
Thank you very much. My specific question was, like, if you have a clinical trial, like for a drug that's approved, right? Like, so you can say part of this was done by AI and that made it come to FDA approval faster. You know, this was a drug for cancer or cardiovascular or any other drug, right? Like that have gone from preclinical to clinical development. But the steps that you mentioned are definitely very much emerging to incorporate AI in clinical trials, and thank you for that. With that, I would give my co-moderator a chance to bring the questions from the men's side.
Ji, Assalamu alaikum. Are there any questions for any of the presenters? Okay, so I'll go ahead and start. You know, kind of. So I got pretty excited to hear about Dr. Salman's presentation. I'm an engineer myself, but I come from a very traditional form of engineering. You know, brick and mortar engineering. We are starting to use sensors in our daily lives. I'm in the water business, so we do employ sensors, but they're very crude sensors. So questions for Dr. Saab, and when he mentioned fluid mechanics and thermodynamics, it you know, perked my ears a little bit. I'm very curious. These sensors, I mean, you know, they are very sensitive. They gather a lot of data. How do you, you know, how do you? What's the plan for the improvement of these sensors and refinement of data so that you can really pinpoint or actually improve the quality of data that you get for better decision making? If that does that, Dr. Saab, do you understand what the question is? Yeah.
Sure, Jazakallah for the question. The algorithms have evolved over years. This actual technology is well over 10 years old. And we initially tested it on elderly patients in a nursing home, which is actually, we call it the Elder Care Lab, which is actually owned and operated by the University of Missouri. And so nursing home residents volunteer to become test subjects. And we started there. But this is why the source of truth is the arterial catheter and the waveforms in the arterial catheter, which we then record and timestamp in real time with this sensor. And it's the algorithm, the MATLAB algorithm that they use and refine, and the filters that they use to filter out the noise that has evolved over time. But the key is a source of truth. Now, you could argue, now notice I said truth and not fact, because truth is relative. Some could argue, what is the real source of truth? Is it the arterial catheter? Is it an ultrasound of the heart? Right? It's all relative. So I think over time, we're learning more about kind of what we can gather. I'll tell you how, there's one story that's actually how frighteningly accurate some of this is. In the initial tests, we compared the arterial waveform data, date and time stamped, with the BCG sensor data. And an astute engineering student on the study noticed there's something off about it. And she looked at it and then decided, what if I offset them by an hour? And then when she moved one of the tracings just by an hour, they lined up almost perfectly. And what she realized is the daylight savings time that we had set for the arterial catheter was off by an hour. We forgot to reset it. So in fact, the time was an hour behind, and the data was so accurate she could realize that in fact you just switch it for an hour, we just discovered a very accurate clock. So that's just an example of the evolution of the sensor data.
Jazakallah. Any other questions from the audience on men's side? Ji, Ahmed Khan Sahib.
So the Quranic verse you mentioned early in your presentation was that God says, I'm closer than your artery. And how do you, in your presentation, how do you reconcile the two? I mean, just for laymen like us, where do you see that fit into what you see in trial data?
Jazakallah for the question. So I used it for two reasons. One, the first part of that verse were the whispers that the soul speaks to itself. And I see that as clearly Allah Ta'ala has created a very complex, one of the most complex creations in the universe that is modeled by everything else by a mathematical model. And just as the universe and the planets are governed by mathematical laws, so is the human body. And I figured our mathematical model, which we use to reconcile that signal, is just telling us what the Creator already knew, which is, yes, these are the mathematical principles governing your cardiovascular system, and only now did you discover what the method is. It's not just your heart rate and your blood pressure. And the idea of being closer than the jugular vein means that clearly there is a spirit governing this mechanism, and we are just scratching the surface as to how deep and complex and simple that mechanism is. So that's how I would reconcile.
Jazakallah. Dr. Saba, we don't have any additional questions on this side.
Okay, Jazakallah, we have question here from Aziza, and she'll ask her question now.
So it may sound very simple, but I'm sure that you can explain it. How do you distinguish in normal body motion, like if, even though it's a sedated person, but there must be some motion that the patient is making? How do you distinguish that from, how do your sensors distinguish that from the other signals that you're interested in?
Sure, Jazakallah for the question. So so clearly there'll be a lot more noise if I were moving with the sensor, which is why the sensor has been tested on people who are fairly motionless. And the sensor over time can detect differences. They have filters. So the MATLAB algorithm has filters that can detect things at a certain frequency, certain amplitude. And motion, body motion, is a far different frequency and amplitude than the subtle motions of the cardiovascular system. That's probably the simplest explanation. I did not write the MATLAB algorithm. I'm not going to take credit for that. That's clearly why we have a multidisciplinary engineering team that does it. But as I understand it with the engineers that I work with, it's the filter designed to do it. And so one of the ideas that I had is you could have a separate sensor that just sits on the body that records body motion, and essentially design a filter that filters out body motion as you reconcile the two signals. They're able to do it with one signal, with one sensor and 3 axes, as we understand the development. But that was my first question: how is this going to work on people who are moving? So right now, it's designed for people who are motionless. And one of the applications that I was hoping for in the future is using it in the neonatal ICU. So premature infants that are relatively motionless, sick in a kangaroo bed or an incubator. Another non-invasive way to assess. But again, you just have to design the filter for that particular patient population, which we have not done yet.
Anyone else has a question?
Jazakallah.
We have a question, one more question.
Gigi, please.
So my question was about like AI and healthcare and like the trials, because like today, like when the AI that we use, we don't really trust it to like do the right thing or say like the factual information. So in healthcare, how much do they really like trust AI to like do things correctly? Or if they don't trust it, then how do they make sure like that they don't do the wrong thing? How do they fix that or correct for it?
Thank you. That's a great question.
So that's why there was a disclaimer at the end that you don't trust AI. You always validate, right? But it speeds up the process, right? And it does. There are things which are not visible to the naked eye. For the algorithms, for example, in case of let's say you're looking at an image, right? And that's why I was saying that Dr. Rafi would explain it brilliantly. The AI will be able to diagnose show you the boundary of the tumor, but it does not know whether that tumor is malignant or not, right? So it gets you closer to the truth, but you can never trust it. It is the model of machine and human. You cannot take the human out of it. So especially with such critical in such critical scenarios, you do not trust it blindly. It gets you closer to the answer, but you still have to validate.
Thank you. We can give it to your side. now. You have
G,
any questions?
There is Hamad Sabri, you have a question? Okay. I think we have a few minutes. So just again, question for Dr. Salman. Again, it's very inspiring that he started off as an engineer, as an aerospace engineer, and then he transitioned his career into medicine. Dr. Saab, we have, mashallah, quite a few young people in the audience, both on men and women's side. This is really inspiring. You know, with the work that you're doing, it's very cutting edge. It's non-invasive, which is amazing. What sort of message do you want to communicate to these our up and coming young professionals or students? How do they prepare themselves for a career, for like a fulfilling career like yours? Alhamdulillah.
Zaga, thank you for the question. The first thing I would answer is, don't follow me, right? So have a career of your own, and don't be afraid to pursue things that other people don't think are worth pursuing, or ask the questions that people don't think you should ask. So medicine in particular, and as a former engineer, I still feel like an outsider, and surgery especially can be very stubborn and very algorithmic, where you are afraid to ask questions. But if you ask enough questions and you have enough faith in your training and you knock on enough doors, I mean, it's taken me, I'm going to date myself. I graduated from medical school in 2001. So it's taken this long for me to find kind of a kindred group of people that have the same interests and kind of same, background that you can work with, right? Most of my partners have no idea what I'm doing, and they don't understand it, and they're just surgeons at work. So have the faith in your training and just find enough people. If it means even moving jobs, right? This is my third job. Don't be afraid to kind of push your own boundaries and your own envelope. So, I mean, I could have just become an engineer and just worked as an engineer, but I just knew something was wrong. I wanted to do something different, something, that we all have heard. And Hazur, Saab says to us that you need to do something different, change your life and your career. And that's kind of what I did. So have the courage to do it, and don't be afraid to ask the right questions and find the right people, because you may feel like you're the odd one out in medical school. Most people are not engineers. Most people don't think like engineers, for better or for worse. So you're going to be different, but you're going to have a lot to offer. And in fact, there are certain residency programs, and even in medical school, very few, especially some in Texas, that are intentionally creating training programs to combine engineers and physicians and scientists together as part of your training program to do just that, to leverage those multiple experiences so that you can have a far better think outside the box system at solving problems. Because clearly they know the old system doesn't work, and we and our Saab here and our moderator are kind of part of that new system. So don't be afraid to kind of push the envelope a little bit.
Jazakallah. Dr. Saab, I think we're good, unless there are any other questions.
We have a question here from the ladies' side. So,
Assalamu alaikum, mashallah, wonderful presentations and great discussion, learned a lot. I have one question for Sabahat. Great talk, actually. We all know there's so much side effects from the chemotherapy, and we know vaccines in general don't have much side effects, but do you know of anything particular to these neoantigen vaccines you think that could be potential side effects? Jazakallah.
Assalamu alaikum, Jazakallah for the question. So because neoantigen vaccines are an immunotherapy, I would say maybe the most concerning side effect that people think about is immune-related adverse events, or essentially overactivation of the immune system. And so the last thing we want with neoantigen vaccines is to induce too much T-cell activity and too little regulation. That would cause too much killing of both tumor cells and also potentially healthy cells. So there are some incidences, of course, and that's why this is still limited to clinical trials, where a patient's immune system essentially becomes hyperactive post-neoantigen vaccine treatment. However, again, relative to other therapies, this is relatively low. And in fact, with neoantigen vaccines, I think potentially a bigger problem is the vaccines not being effective enough and not inducing enough of a T-cell response. And this is due to a number of factors, including poor delivery of the neoantigens, poor uptake by cells, and so on. So it seems like actually, rather than side effects, we may not be inducing enough therapeutic efficacy. Jazakallah.
Thank you, Sabahat. That was a great answer. I think it explains the role of neoantigen vaccines versus conventional vaccines. I just had a follow-up question on that. How do you see these neoantigen vaccines? Do you see them as standalone, or do you see them as combination therapies with some checkpoint inhibitors or other standard of care therapies for cancer?
Jazakallah. So, in fact, most of the clinical trials, I would say, involve some sort of combination therapy, because we have seen with cancer that combination therapies do better than a single treatment. And as I mentioned, in the neoantigen vaccine delivery itself, often adjuvants or additional immune stimulants are included even in the vaccine delivery. So that's another form of, I guess, combination therapy, or including things other than neoantigens. And finally, and importantly, one big thing that's tested in clinical trials is even number of dosages, for example, and an appropriate dosing schedule. So even neoantigen vaccines, people are starting to realize that maybe one dose isn't enough, and so that's also something that's tested.
Thank you. I just have one question for Dr. Salman. Again, very fascinating talk. And one of the questions is, did you see any differences in the types of patients with these sensors, meaning some with chronic heart disease versus some with mild? And how did the sensor respond on those? And did you gather enough data for making consistent conclusions?
Talked about the question. So, at least in our ICU, we were only able to test this prototype on about 6 or 7 patients, and right now we're actively enrolling more. Other than some gender differences, right now the sensor and the studies that we're doing have not gotten to the point where I can separate somebody that has heart disease, heart failure, or different things, because I think the sensor alone won't be enough. The sensor combined maybe with EKG data will give us kind of more information, especially on the timing between the EKG. That's going to tell us a little bit more, and that will allow us to generate that PV loop. And once we do that, I can get a lot of information from the PV loop, because PV loops that are done now require an invasive catheter done often in the animal lab, and some human patients get them, but they're very sick. And that's the only way to get that source of truth. So we are not there yet, but that is the goal eventually. Thank you for the question.
Thank you. So we would like to now conclude this session. We are at time, and our next agenda item is lunch. And then we have Juma or Salat. So with that, I would like to thank all the speakers for this great discussion today, and also the co-moderator. And I would share my mic with our co.
Jazakallah.
Do you want to say a few conclusive words, Junaid, Malik Saab?
Jazakallah. No, this has been very inspiring again. I think these kind of gatherings, we kind of get to learn about all these different topics, and I think I always look at the audience, and mashallah, we have a lot of up-and-coming stars and a lot of leads that they can look up to, and so this is Jazakallah to all the speakers for sharing their knowledge. Jazakallah. Assalamu alaikum.